All right. So, just start class by talking about what I like to call some false dichotomies. And I'll be honest, I blatantly stole this slide um, off of a professor at University of Florida who taught a psychiatry class. And I really like this slide. And think it's a good way to start talking about this. First of all, is the idea of mind versus brain, where neurological illness is perceived as a disease of the brain, while psychiatric illness is perceived as a disease of the mind. Or, in other words, where you look at the psychological versus biological, that some mental illnesses are purely psychology, while some are purely biological. This isn't the case. There isn't a pure psychology. There isn't a pure biology. There really is no differentiation between a brain and a mind. They work together. They work in concert with each other. These concepts that we have, basically, we need the biology to create the psychology. And for every psychological process, there is some biological underpinning to that. So these are not two distinct things. Your biology can affect your psychology. Just having, even things that we don't necessarily think of. Heart disease. There's a high comorbidity between heart disease and depression. And there's a physiological mechanism going through your immune system, the cytokines that go through that actually do cause depression. Of course, stress and what you have, or changes in things that would cause fluctuations in hormone levels. There are things in biology that can change that will absolutely affect your psychology. And your psychology can affect your biology. Outcome results. Now, whether we're talking about behavioral actions, such as when your doctor tells you need to lose weight, or your doctor tells you need to stop smoking, or you need to stop eating fatty foods, or what have you. We see all these diet pills on TV. They all work. Almost every one of them has a scientific underlying idea of we block this chemical, we raise this chemical, we block this receptor, and this is something that has something to do with feeding satiety, and therefore that should make you stop eating. But the thing is, they don't work. They should theoretically work, but they don't, because what those pills don't do is they don't affect your thinking about it. There's so much about how we eat that is based nothing on the neurochemical signal my stomach is sending to my brain, but has to do with I'm driving home right now, I'm late, my kids are going to be hungry, and gosh, should I run through the McDonald's drive-thru and just pick up some hamburgers really quick, or do I go home and cook for the next hour? And no diet pill is going to change that. So you can't really separate out psychology and biology. Because for everything, okay, well, this has something to do with psychology, that psychology is going to affect your biology. It is going to change you. There's so much in there about what social support will do, that positive benefits from therapies will do. So they're not really two separate things. They go hand in hand. And this goes into always the genetic versus environmental, nature versus nurture. We always like to talk about that in psychology. It's a stupid thing to really talk about. There isn't a nature versus nurture. There will be one thing that we will discuss in this class that is a 100% genetic disease. That's Huntington's disease. Everything else we'll talk about has some genetic component to it and some environmental component to it. Look at something like schizophrenia. If we look at identical twins, share 100% of the same genes. If one person has schizophrenia, 50% of the time the other identical twin will have schizophrenia. Okay, so if it was 100% genetics, then they should be 100%. If it had nothing to do with genetics, if it was purely environmental, if we raise those two twins separately, then there should be a 0% or 1%, just the rate of the general population. So they both combine. They're both important. You have a genetic predisposition for something, and then something in your environment then triggers that. Whether that's something in the environment is a psychological stressor, or whether it's a disease that's contracted at just the right time during development. But something else comes in. So it's really a false dichotomy to talk about these things as being separate. They're all together. They all work in concert with each other. The biology and the psychology work together. Now, where does this field come from? Well, if we look back to Hippocrates, so we're 400 BC, this is the description that he gave of the brain. Men ought to know that from the brain and from the brain only arises our pleasures, joys, laughters, and jests, as well as our sorrows, pains, griefs, and tears. Through it in particular, we think, we see, hear, and distinguish the ugly from the beautiful, the bad from the good, the pleasant from the unpleasant. It makes us mad or delirious, inspires us with fear, brings sleepiness and aimless anxieties. In these ways, I hold that the brain is the most powerful organ in the human body. So, 400 BC, that is as good of a description as you can give on the function of the brain that you could give today. So he nailed it, absolutely. But not everyone agreed with him. If you look at Aristotle, he thought the brain acted as a radiator to cool our blood. In fact, he put that into the heart. The seat of the soul and the control of voluntary movement, in fact, the nervous function in general, are sought to be in the heart. The brain is an organ of minor importance. So there was early debate about what exactly the brain did in the body. Now, if we look at Galen, there's one of those History Channel type specials on him one time. They call it Galen, Doctor to the Gladiators. One thing that Galen did is he was, in fact, a physician who worked on patching up gladiators after they'd hack each other apart. And one good thing that this kind of did for him is basically the gladiators would dissect each other for him. And so he could, kind of look up in there and see what was going on in the body. He also did dissected animal brains. And so what Galen did is basically he used empirical observation. Instead of philosophically saying what he thinks the brain does or what the heart does, he looked. He used observation. And what he noticed when doing dissections in animals was that all the sensory organs were connected to the brain. And so therefore that led him to believe that then the brain must be what is organizing and integrating all that sensory information. So now we're using an experimental method in a way to determine what is going on in the brain. Okay, so nothing happened for the next 1,700 years. So we're going to skip to Descartes. Now, we always try and use whatever the latest, greatest technology is as a way to describe the human body. Probably if you took cognitive psychology, you probably at some point used the computer to describe the brain. Your short-term memories like RAM, your long-term memories like a hard drive, that type of thing. And, this went on, describing the human as a clock, describing human what have you. And so one of the things that was really going on in this time was hydraulics. And the story goes that they're in the royal gardens in France. They had these statues. And you would walk through the gardens and you would step on a stone. And that stone would cause a transfer of fluids. And that would then cause a statue to move. So it was, 17th century Disney World, with animatronics. And Descartes was evidently very impressed by this technology and sought parallels in that in the human body. And in fact, what they thought was that in the brain you have these cavities called ventricles that are filled with fluid. And you know what? When you flex your muscle, it kind of gets a little bigger. And maybe that's just maybe you're transferring fluids. Maybe you're using hydraulics. And you're just simply transferring fluids and filling up the muscle there, causing the movement. So that's not how it works. But that was thinking. Now, one thing that Descartes did get right was that reflexes are too fast. Reflexes don't make sense. I touch something hot and I jerk my hand back. Now, what he ascribed that to is he said, okay, when I touch the hot stove, that the heat from the stove is giving my body energy, and that my muscles are reacting from that energy. Now, that's wrong. But what he was right is that we don't think about that. I could chop my head off, theoretically, and my arm will still do this. Because the reflex actually all happens just in the spinal cord level. So the fact that he noticed how fast that was, and it didn't make sense that our brain was involved in thinking about it, actually was a correct way. Now, he argued that there was a separate mind and brain, and that this interaction between the two occurred very specifically in the pineal gland. Right here in your brain, there's your soul. Why do you think that? Because almost everything in your brain, there's two of on the left and the right-hand side. There's only one of these, so it must be really important. Why is it called a pineal gland? Looks like a pine cone. Okay. Now, really, for the most part up to this point, most people were approaching neuroscience in a philosophical way. But then we started having advances in technology that allowed us to start looking at the nervous system in a completely different way. And one of the biggest advances of this was the invention of the microscope. So as soon as people started inventing the microscope, people started sticking things under it. And they looking at it big. And of course people started sticking tissue underneath the microscope. And what they discovered was, hey, there seem to be these little blobs that our tissue are made up of. They're made up of cells. And so this was a big discovery. This was something, that we couldn't see without the microscope. Except for when you put nervous tissue underneath the microscope. Nervous tissue didn't seem to have cells. So the theory went that our body is made up of cells except for our nervous system. Then came along Golgi. And he developed what we call now the Golgi stain, or the silver chromate stain. And basically this was a way of prepping tissue. This is a way of putting dyes and solvents and what have you on tissue. And for some reason, and we actually to this day still don't know exactly why this happens, only a small percentage of the cells will take up this stain. So what you get is something that looks like this. Because since only a small percentage of the cells will take the stain up, you can start to see them on a background of tissue that did not take up the stain. And so when we did this, we discovered that, yes, in fact, the nervous system is made up of cells just like the rest of the body. The difference is that the cells are so tightly compacted that they're not easily distinguished unless you can do something that then highlights certain ones of those cells. So Golgi developed this stain, and then Cajal then started looking at these cells in the nervous system. And what he started noticing was that, basically that's what he was just looking at, cells after cells after cells. And he noticed that the cells in the nervous system look different than the cells that we see in the rest of the body. They have a different shape to them. And so here I have just kind of the cartoon version of a generic neuron, which are one of the basic cells in the nervous system. And you can see that there are certain parts of a neuron that are specialized for the neuron. Now, like almost all cells, you have a cell body and you have a nucleus. We'll talk about this in detail later. But coming off of this, you have these things called dendrites. And then often you have one long process that then goes and then connects with something else. Another neuron, a gland, a muscle. And so Cajal, just by looking at slides under a microscope of neurons, developed what we call the neuron doctrine. And he said, okay, the way these cells look, we know that the nervous system does a lot of communication, and so it makes a lot of sense that the neuron is responsible, that the neuron must be the basic signaling unit of the nervous system. And just by looking at how it looks like information would come into the dendrites and then would be transmitted along the axon to its target, to where information, it would talk to other dendrites, send information on, and send it that way. So he got all this just by looking at it. He just said, this is what makes sense. And it turns out he was 100% correct. This is exactly how neurons take in and process and send information. The information comes into the dendrites, travels down the axon to its target. And for this work, and Golgi and Cajal won their Nobel Prize for that work at the turn of the century. Okay. So that's the cell physiology world, or cellular anatomy world. What's going on in the field of physiology? Well, Galvani in the 1700s, he determined that electrical stimulation of frog nerves will cause them to contract. So this is the basic idea. He could just take electricity and he could cause muscles to move with this. So that gave this idea that our movements, at least, are essentially an electrical signal. Now, we're not purely electrical, but it is an electrochemical signal. Now, Du Bois-Reymond, he discovered something called the resting membrane potential. And let me see if I can. In an experiment, what he had is basically, if you can imagine like a. That doesn't. If you can imagine a small dish, and if you put a piece of tissue in that dish floating in there, and then what he did is he put an electrode recording inside of the tissue and then outside. And so basically he measured the difference in the electrical potential inside the tissue and outside the tissue. And what he found was that inside the tissue is negative in relationship, carries a negative charge or a negative differential than the outside of the tissue. So what this is doing is this is setting up the idea of that we do have a electrical, if you will, way that our body can communicate, that we have this differential, that there is a difference in the charge potential between inside and outside of tissue. So if we want to know how can electricity possibly cause movements, well, we now know that there is a difference in the electron placement inside and outside tissue. And we'll talk about that in more detail later. We'll talk about why that is. But we'll see that this is a very important aspect for how our neuron, if we didn't have this, resting membrane potential with a negative charge to it, our neurons would not be able to communicate. We would not be alive. Mueller, the early 1800s, he developed the doctrine of specific nerve energies. So you can ask the question, how come I see visual things and I hear auditory things? Why don't I see sounds? Why don't I feel how something tastes? Is there something unique in my brain about the information coming in through my eyes that's coding visual information versus something that's coding auditory information? And the answer is no. Your neural signal is a neural signal. There is nothing different. We'll talk later about what's called action potential, and that's how the neuron communicates information. And there's nothing different about an action potential being sent from a visual stimuli versus an auditory stimuli versus a tactile stimuli. They're all the same. What's different is the pathway. The reason when I touch something and I can feel this little ridge right here is because my finger is touching it and it's sending information to the part of my brain that decodes tactile information. When I'm looking at your yellow jacket, the reason I can look at that and I know that's a yellow jacket is because that information is coming through my eyes, is going to the part of my brain that decodes visual information, and then is going to the parts of my brain that can identify yellow and can identify a jacket. So the function of a neuron is determined by its pathway. That's the main take-home point. That if we want to know what something does, we just simply need to know what's it connecting to. That there's nothing necessarily overly specialized about the neuron taking the information being transmitted, but it's about where is it starting, and where is it ending in the brain. Now, von Helmholtz, also in the 1800s, showed that nerve conduction is slower than electrical conduction. So basically, this idea that we're all working on electricity somehow. What he's showing is no, we're not a bunch of electrical wires. We don't have electricity going through our body. That something else is going on, and it is, because it's an electrochemical reaction. It's about differences of differences in ions, negative and positive ions moving, not like electricity running through a wire, which is just a stream of electrons. It works in a different way. And while an electrical signal can still simulate that and can still cause a muscle contraction or what have you, that's not the same thing that's going on in the body. So these are things in the field, in areas of science that are converging, helping us understand the function of the central nervous system. But there was a big question about how the brain itself worked. And there were two big theories about this. One theory was the aggregate field theory, and the other was the localization of function. Basically, the aggregate field theory was this idea that your brain worked as a whole. And that there was an individual, just to contrast that to localization of function, the localization of function said that different parts of your brain were responsible for different behaviors or different things that you did. Whereas aggregate field said, no, this brain works as a whole. Now, the localization of function had an early adopter, and that was Franz Joseph Gall, who developed phrenology. So phrenology was this idea that your skull was a reflection of your brain, of what your brain looked like underneath, and that basically by examining your skull and where you got bumps at, or how big your forehead is, or how I could tell things about your behavior. And so you've probably seen the phrenology, pictures, and these, and they could tell you where on the skull all of your different behaviors were. Now, this guy was a crackpot. It was really a lot of excuse for racial segregation. It was really more of these races have these different types of skull features, and that this is how we're going to classify that they're inferior in some way. That having a forehead shaped like this made you a criminal, or something like that. That was really more what this was about. So this didn't do the theory of localization of function any good to have him associated with it. And so basically people, by rejecting phrenology, also were rejecting the idea of localization of function. In fact, there was actually some good experimental evidence for the aggregate field theory. So if we look at Pierre Flourens, he specifically wanted to test out these ideas of Gall. And so what he did is he did research of using rabbits and dogs, and he would, through surgery, would perform lesions, would make lesions in their brain and look for behavioral deficits. Because if Gall is right, and this little area of the brain does this behavior and this little area does this behavior, if you cut that out, then you should see some kind of behavioral deficit. The dog's behavior will change in some way. You will see some lack of a behavior from when the dog brain was fully intact. And in fact, what besides sometimes I'm running into some motor deficits, he didn't see that. There was no distinguishing between the brain-injured dogs and those that weren't. Now, I'll take a little aside right here. Most of what I will talk about in this class, a lot of what I talk about in this class, is determined through similar animal experimentation. We simply can't do this type of experiment with a human. We can't go in and selectively destroy part of their brain and see what kind of behavioral deficits there are. There are people that wait for a human to become injured, In fact, we'll talk about, one of the biggest cases that helped us to understand how the brain functions, exactly that. A person had a very odd neurological symptom, and then that was well documented, and then upon autopsy after their death, they said, oh, they got a big hole in their brain right here. I bet that area of the brain was responsible for that. So we can, after someone who has had certain types of surgery or following certain types of strokes or traumatic brain injuries, we can see these behavioral deficits, but they're random. We never know when that's going to happen. Of course, an injury is never going to be just a clean injury. Animal experimentation is used a great deal. Now, I don't know what they did to these poor dogs at this point, because this was at the 1800s, and we didn't exactly have things like ethics, or I shouldn't say they didn't have ethics. We didn't have laws about things like this. Today, research done today has a very intricate system of laws and regulations and ethics boards that govern the use of animals in research. This came out of the Animal Welfare Act. Is that late 70s? Late 70s, I think, or early 80s, somewhere right around there. You had the Animal Welfare Act, which then set up the system of what we call institutional animal care and use committees, or IACUCs. So any research now involved in anything use of live vertebrate animals would go through an institutional ethics committee that must approve the experimentation that you do. I sit on the IACUC committee for both UHCL and at the UH Central campus. And it's not a simple project, and we almost never approve of projects right away. I could probably, in the 4 years I've sat on UH's committee, which we meet once a month for about 3 hours each time going through these protocols, I could probably count on one hand how many times we've approved a protocol right away. They are, there's a high bar standard. Basically, there's this concept of what we call the 3 R's, where you reduce, no, not reuse, reduce, refine, and replace. So reduce, you want to reduce and use the fewest amount of animals possible for your experiment. So you actually have to do what they call a statistical power analysis to show what's the fewest number of animals you need in order to get statistically significant results. The refine aspect is that you have to use the least evasive methods possible for this. And so if there's a, to use a non-animal example, once my brother had surgery on his knee, they could have done it where they just basically opened up his entire leg and went in, and then he's going to be on a cast and on crutches for the next 6 months. Or he could go to Tulane, because some guy was doing some experimental procedure there that was orthoscopic, and he was walking within about 2 weeks after that. If that exists for an animal surgery, you'd absolutely have to do, no matter what the cost is, no matter what it is, you have to do that, because that's going to be a less painful procedure. That's going to be, we don't care why you have to do that. You have to do the less invasive procedure. And then replace. If there's a way to use maybe this is slightly speciest, but basically use a lower organism you can. If you can avoid using a primate by using a dog, use the dog. If you can avoid using a dog by using a rat, you use the rat. If you can avoid using the rat by using a fish, then you use the fish. If you can do that by using a fruit fly, you got to use the fruit fly. And you have to justify that. The application makes you justify these things about why do you need this. And if you can get away with doing it in a petri dish with just cellular, with just tissue, you have to do that. You have to show why you have to do this in a live animal model. Why do you have to do this on this particular type of animal? And as technologies get better, we can push things to lower. Right now, the mouse is the most common animal research model used. When I was coming through, when I was working in Boston, and I was trying to do sleep recordings in a mouse, it was incredibly difficult. I had to custom build all of my own electrodes and everything. I would spend days and days just building the little parts that I would do to make the sleep recording stuff. Now there's just a company you just order this stuff from. The technology's gotten even better. And then even 10 years before that, there's no way the parts would have been small enough to do it. In fact, they had to do it on a cat. You couldn't even get down to a rat size. And now you're very rare to see, people doing dog and cat research, they almost don't exist anymore. Whereas in the 80s, that was because we've gotten everything so small now. And it's difficult to do research with the dog. Plus, people don't like that. I'll be honest, I will not do that. I would not be part of a research team that did research on a cat or a dog. Not because I don't value that research. It's just not what I'm going to do. I'll work with rats and mice. Actually, I hate mice. Mice are nasty little animals. Just that they're mean. They bite. I'd much rather work with a rat. But mice are wonderful genetic models. But with just things that we can do over 10, 20 years, the technology has advanced. You can do functional imaging on a rat now. When I was at Florida, you can do that. They got this huge magnet. You stick the rat in there, and you can see what's going on in his brain. Incredible what you can do with an animal now that you couldn't do 10, 20 years before that. And now the stuff that they're doing with zebrafish, I almost wish I was coming through grad school right now so that could be the experimental model, just because I like fish. But, just the stuff they can do with fish now. I don't know if fish is not the best sleep research model, but flies, you can do sleep research with flies. Flies sleep. Anyways, so but what I want to make sure that is that at least research conducted in the past 20, 30 years does undergo very strict ethics approval before it's done, and that this isn't something that's willy-nilly done. I'll give you an example in a little bit of what's not allowed nowadays. But it's the same thing with human research too. You'll take social psyching, you'll discuss Milgram, and you'll discuss the Stanford prisons experiments and all these kind of things, and experiments where it'd be very hard to get through IRB approvals today. And it's the same type thing. Not everything would you see now. These are still methodologies used. We've gotten much better at this, where he most likely had to open up the skull and probably took a knife and cut out a piece. Then progressed into that someone could put an electrode in there, turn up the voltage, and kind of burn away a little piece of it, which now is now these very highly specific toxins. I'll show you what a modern example of a lesion looks like. This is a study I did several years ago where I lesioned an area of the brain called the ventrolateral preoptic area. And basically you can see all these little purple dots, those are neurons. And so you can see in these areas where you see very few neurons in there. So I didn't go through and cut away with a knife or go burn away with, didn't burn anything away. In fact, what I had was I took a small little pipette, placed it into the brain, and injected a toxin that was attached to another chemical that would bind to receptors of neurons and would selectively kill off certain neurons. So that the brain is still left intact for the most part. It's just in this particular region, I have it's not 100% because this isn't 100% die-off of neurons, but this is anywhere from an 80 to 90% die-off of neurons in that region there. So this is an example of how these techniques get better over time. All right. So Florence, he does these experiments. He does these with lesions in different areas. Says, hey, I can't tell any difference in their behaviors except for some motor behaviors. So therefore, Gall's wrong. There isn't that this part of the brain does this and this part of the brain does that. And Carl Lashley, turn of the century or early 1900s, was doing the same type experiments. He was running rats through mazes. So he trains a rat to run through a maze. He selectively makes regions in different parts of the brain and found that no matter where he went and cut in the brain, it didn't impact their ability to run through the mazes. Now, what he did find was that the bigger lesion or the bigger chunk of brain he took out, now that would cause a problem. So it wasn't where he made the lesion, his conclusions, wasn't where he made the lesion, it was how much of the brain he destroyed. Little bit of brain damage, no problem. Middle bit, middle problem. Large brain damage, large problem. So this all fit into the idea of the aggregate field. That the brain was acting as a whole. It didn't matter where the lesion was occurring. It only imagined how big of an impact you were making into the function. Okay. So, we got some evidence that the aggregate field theory is okay. How about the localization of function? Getting beyond phrenology, what other evidence did we have for it? Well, Charles Sherrington studied the knee jerk reflex. So this is the classic, sitting on the doctor table, they hit your knee kicks. Anyone know why? Why does that happen? How come if we hit your knee, your leg kicks? And so he started studying the neural mechanism of this. And what he showed is that there are receptors, neurons, that are located in your knee that sends information into your spinal cord. So this is a segment of your spinal cord. Imagine a person laying on their back, and we just took a slice like this. So we're looking down at a section of spinal cord here. And what we saw is that the neuron had sensory information came in to it and then communicated with a motor neuron that then sent information back to the muscles and caused them to kick. So in the spinal cord, you had a sensory area and you had a motor area. An area of the spinal cord that was responsible for sensory integration, and an area that was responsible for motor output. This comes back to the Descartes part. Notice there's no brain in there. This can all happen in your spinal cord. Yeah, there's information that then goes up to your spinal cord, but not necessary. Okay, so he won a Nobel Prize for this research. So the idea for the take home from this is, okay, well, if our spinal cord, if you ever looked at a brain and ever looked at a spinal cord doesn't look like much. There's nothing much to it. Our brain just looks complicated. It's got all these things sticking out and bulging and parts of it here and there. So you just look at it and you go, that must be a complicated organ. You look at your spinal cord and go, this must be one of the simpler things in our body. If your spinal cord, though, has a localized function, if there's a sensory area to your spinal cord and a motor output area, then why would we assume that your brain was any different? If the simple part of your nervous system has localization of function, then it would make sense that the higher parts of the nervous system probably also in some way have some localization of function. Okay, well, how about some evidence in the brain? I'm never exactly sure how you pronounce this. Fritsch and Hitzig. Now, here's an example of what would definitely get you thrown in jail now. So, the story is they, on their kitchen table, would, knock a dog out with ether or something, and then we go around and start poking its brain and see what would happen. Now, it is important to mention that your brain does not have any pain receptors in it. I can poke around in your brain and you won't feel it. It doesn't hurt. If we just use lidocaine and kill your, the sensation of the scalp you got to go through. In fact, when they do brain surgeries today, you are awake during it. They need you to be awake because they're going to need to talk to you while they're doing this. Because I said every brain's different. Because they're not going to know, and they're going to want to really avoid it. They need to go in and remove a tumor, or they need to go in and remove an area that's causing uncontrollable seizures or something like that. They're going to try to minimize as much functional damage as possible. If they put you out, go in there, cut out a part of your brain and wake you up, they don't know what they're going to get. So it's much better for you to be awake during this, and for them to be able to start poking around the brain and say, hey, what's happening? I mean, they do it in a more detailed way. Right, exactly. They press something, all of a sudden say, I can't see out my right eye. Okay, stop pressing that. And so they'll have somebody, once again, they do it in a more sophisticated way, but there'll be somebody there doing basically kind of like cognitive testing with you while you're there as they map out parts of your brain. And they'll figure out, okay, this is important, okay, nothing seemed to happen here, this is the place we can go in. And so that helps minimize the damage that they do. So anyway, so what they did is they stimulated with electrodes different areas of the dog's brain, and they discovered what's known as the motor cortex. So basically it's this strip of brain here that controls your motor movements. And what they found was if they stimulate along this strip, the dog would do something. They press and they stimulate with the electrode and the dog would move in some way. And they found that different parts, as you move along that strip, different areas would move. So, and they discovered that it would be a side on the opposite side. So they stimulate over here. They stimulate on the right side, your left hand moves. Left side, right leg moves. And that's a concept that's going to actually turn out to work through almost everything in your brain. That your right side of your brain controls the left side of your body, left side controls the right side. So, they've shown a clear demonstration of part of a localization of function that, at least for motor control, there is a specific area of the brain responsible for motor control. All right, how about an example with some humans in it. So, Broca. Broca was studying people who had trouble producing speech. Specifically, he had a patient that we called taw, because this patient could only say taw, 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 taw. Huh? Taw, 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 taw. Actually, I have my wife's grandfather, after he had a stroke, and he actually has this exact same thing, and he's cognitively quite intact. You play cards with him, he will beat you every time. He was a former Air Force pilot, was an engineer for a while. I mean, he's still got the sharpness to him, but when he talks, he just wah 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 wah. You know, no matter what, wah 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 wah. You know, that's just he can't form words. And there's nothing physiologically wrong. There's nothing wrong with his mouth, his tongue, the ability to form speech. That's not what's wrong. This is a neurological problem. So no physiological problems that explain why the speech can't be produced. And so we call this Broca's aphasia or expressive aphasia. And so this can come in different degrees. So the patient taw, or like my wife's grandfather, that would be the extreme case. But you can have milder cases of this where the person just simply has non-fluent speech. It's slow and it's halting. They have difficulty articulation or slurred speech. They have problems with grammatical devices such as verb endings. Their comprehension is fine, at least normal comprehension, but you can still trick them up with certain very specific ways of asking them questions. And so, after death, after the patient passed away, Broca then did an autopsy. And what it showed was there was a very specific area of the brain that was damaged. We now call that Broca's area. And this area is very important for the production of speech. And so if this area is damaged in some way, by disease, by stroke, by injury.
Does it have to be damaged, or can it be maybe born? I mean, just born that way, or maybe it happened during.
I'm sure there could be a way that through birth that problem happens. I'm sure that could. When the congresswoman, I forget her name, was shot. Yes. I knew instantly when they described her problems that she was shot on the left side of her brain. This is, the difficulty she has speaking now. This is classic that this was an area that was affected. Now, she's gotten better. That's the wonderful things about neuro rehab that we have now, that we can do things about this. To give you another example, Carl Wernicke, he was also studying people who had speech problems, but a little bit different. So Wernicke, instead of studying people who had trouble producing language, these people had no problem whatsoever producing language. It's just it didn't make any sense. Can I see your book for a second? Your book has an example. Where is that? Yes. So, this is a person who has Wernicke's aphasia. The person asked interviewer, what kind of work did you do before you came to the hospital? Patient's response: never now, Miss Oiga. I want to tell you this happened when he rent. His Kel came down here and he got rin something. It happened, and these ropiers were him with his is friends like was, and it just happened so I don't know. He did not bring anything around. He did not pay it, and he wrote in all these arranging from the pedis from Ispesed in these floors now, and so he hadn't in round here. Okay, so speech production is fine. The person can talk. They can talk fluidly. So it isn't. That's not the problem. They're still using English words for the most part. But the problem is it doesn't make any sense. So you see excessive and rapid speech, uninhibited flow, but the speech doesn't make any sense. And the idea, of course, it's a little hard to tell whether or not the person knows what you're saying, whether or not what their comprehension. They feel pretty confident that the speech comprehension is poor. But then on the other hand, we don't know what they're saying to know if they, you can't say, can you repeat back to me what I said? Because it comes back out. And the person does seem to be unaware of, they don't know that they're speaking gibberish to you. And so this, there's another area of the brain, which we call the Wernicke's area. And it seems to be responsible for speech comprehension. So what we seem to have is if, hey, it's working. This little thing's animation's never worked before. Weird. Okay. So if I was to ask you to read for me, so like when I was just up here, I had the book and I was reading. What's going on here is I have visual information coming in, so we're missing a little part of this here. So I have my eyes are here, and so visual information comes into the eyes. The eye is going to send information to the back of your brain, to your primary visual cortex. Then that's going to send information to Wernicke's area. And this is where it then makes sense of the words, speech comprehension. And then that's going to send information to Broca's area. And that's going to be your speech production area, who will then send information to your motor cortex, which will then send information to the parts of you that's a mouth, that then produces the speech. And then if I was doing this from the written, if I was doing this from hearing, if I said something to you and asked you to repeat it, what would happen is that we got your ear here, and that information would come from your ear, would then go to the areas of your brain that deals with understanding auditory information, and then go to Wernicke's area, then to Broca's area, then to your motor cortex, which would then go back down to your mouth for you to speak it.
That was actually a pretty decent ear. So.
What's that?
That was a decent ear.
Oh, thank you. So, actually, I want to show you a short little video clip on this. Oh, there. So, I'll show you a fair number of video clips throughout the semester. They're all old. I'll tell you when something is older than... I'm only going to show you the parts that are still relevant, but of course their dress is old. But they still... They want to charge a lot. They still sell these video clips for like a couple $100. They're just really expensive, and so until they just are saying wrong information, I'm going to keep using them.
While the whole brain normally participates in communication, most of what we think of as language depends on the left side of our brains. The area for language on the left side is more densely wired and even structured differently than the same area on the right. In 1861, Paul Broca discovered the left hemisphere's role in language. This brain changed the course of neuroscience. It belonged to a stroke patient nicknamed Ta. He was given that name because Ta was all he could say. After Ta died, Paul Broca examined his brain. Because Ta could not speak, Broca concluded the damaged part of the left hemisphere was responsible for language. It's now called Broca's area. Ta's brain was recently rediscovered in a Paris museum. Scientists x-rayed it with a CAT scan. The black area in the left hemisphere is where the damage occurred. We now know that Broca was largely right. Damage to Broca's area does disrupt language. Until his stroke, Charles Landry had a thriving legal practice. At age 45, a blood clot cut off circulation to part of his brain and put an end to his professional career. At the heart of practicing law is a facility for language. Landry's stroke affected both his ability to understand language and his ability to express himself.
Contingent remainder is sell it, sign it, deliver it, or anything subject to the contingency.
When we talk about language difficulties, we mean problems with either grammar or the choice of words. And if you were listening to him, someone might say, well, his grammar's all right, and even though he takes a long time to find the words, he tends to find them. His difficulties are really all mechanical ones. He's slow, he's got trouble getting going, but none of this is really language disturbance. Well, I think that problem gets resolved very quickly when we start to examine his understanding of language. Do you ever have any trouble with what I say?
No.
Understanding it?
No.
Well, suppose you wanted to communicate with a person who was in a distant city. What apparatus would you utilize?
Telephone.
Very good. All right. And I deliberately made that sort of a curious sentence just to show how well you did. All right. Now let me ask you, do dogs fly?
No.
No. And how about this? Do submarines usually fly?
No.
Mm-hmm. How about a Zeppelin? Can a Zeppelin fly?
Yes.
Oh, okay. I couldn't make the question complicated enough in terms of the fanciness of the vocabulary to throw him off. He immediately understood. And yet, when I produced a sentence that most of us would have thought was about as easy an English phrase as you could create, he failed totally. You know what a leopard is?
Yeah.
You know what a lion is?
Yes.
Right. The leopard was killed by the lion. Which animal died?
I don't know.
That's hard, is it?
No. I don't know. What animal died? I don't know.
You don't know what animal died.
Right.
Obviously, that's got to be an error in understanding the grammar. Now, there's a very easy way to understand it, because if you think of those sentences with the grammatical words removed and try to figure out their meaning, you would have the same problem.
Landry hears all the sounds of the grammatical words, but in his brain, the sentence is processed something like this. He understands only nouns and verbs.
This pattern of having this special kind of understanding difficulty, which is confined almost exclusively to these little words, to the grammatical words, and to the endings, is something that we find very frequently in the patient who's got damage to Broca's area. So the thing that's so perplexing in Mr. Landry is that he doesn't have this difficulty in spontaneous speech, he doesn't have this difficulty in repetition, but when we come to the comprehension tests, he has this difficulty in the most dramatic fashion.
Broca's discovery was important, but it wasn't long until other investigators discovered additional areas in the left hemisphere that contribute to language. In 1874, the German scientist Karl Wernicke identified a third language area in the left hemisphere. Broca's area in front plays a major role in speech production and grammar. The strip in the middle controls and coordinates movement, but Wernicke's area is involved both in the formation of what is said and in comprehension. When it's damaged, speech seems fluent but doesn't make sense. We now know how we process and repeat words. First, sound travels as nerve impulses to Wernicke's area, where it's analyzed, to Broca's area, where sounds are assembled into sequences, and finally to the motor cortex, which directs movement and sends signals to the speech muscles. Charles Landry's brain damage has produced difficulties that are fairly typical of patients with damage to Broca's area. From patients like him, we have learned a great deal about how the left hemisphere decodes, processes, and produces language.
So if you haven't figured out by now, localization of function, they're going to win. We do have specific areas of the brains that are responsible for specific functions. But then how do we go back and explain Lashley's work? You know, Lashley wasn't somebody back in the 1700s doing things. He was actually doing fairly modern techniques. But still, he didn't find that making specific lesions in the rat brain impaired maze learning in the rat. So how can we explain this? Well, think about it this way. I live in Pearland. Let's imagine that before I leave this afternoon, someone tells me that there's a big wreck on 8 and it's shut down right now. Could I still get home? Sure. That is only one way. There are multiple ways that I can work my way. There are some that are less efficient. It's going to take me longer to get home. There are some very roundabout ways I can get, but I can still get there. And basically, your brain works the same way. That just because one particular area of the brain is damaged or one particular pathway doesn't necessarily mean that information still can't be moved around or transferred in the brain. It might mean that the most effective or most efficient way has been destroyed. And let's say for some reason tomorrow they decide to blow up the Beltway. It just doesn't exist anymore. Okay, people are going to complain, and then what they'll do, they'll add lanes onto other roads. They'll make them more efficient. And that's what happens. If you have an area that isn't functioning, then through training, through repetition, we can make other ways that used to be a little pig trail in your brain, we can make them more prominent. In fact, your brain can actually even recruit areas of the brain that never was even involved in that behavior before. I had a friend at University of Florida who did experimental brain damage in a rodent model. And so what they did is they taught the rat to do a reaching task. They'd reach through something and grab a piece of food. And they could put on the motor cortex, they put an array of electrodes. And they could see which neurons were actually being involved in that reaching task. Then they'd go and destroy those neurons. And then they would work with the rat and train it to reach and grab a piece of food again. And what they would show is that neurons that were originally weren't a part of that, all of a sudden, basically, these other neurons would then adopt that behavior. This is the idea of neuroplasticity. Our brain is not something that's just set in stone. We can develop new pathways. We can recruit new regions of the brain. In fact, people who have damage to the left side of the brain, for some people, you can actually get their right side of the brain to start picking up this language functioning. Especially if you're a young left-handed female. You have a better chance of that happening. So, the other problem with Lashley's work is that running a rat through a maze is actually a complicated behavior. There's lots of different ways that a rat can solve the task. Maybe the rat, if you're using a food reward, he can use his olfactory function to smell where the food is. Or if you don't clean the maze well after the rat runs through, they leave little scent trails and they can just follow the scent trail. Or maybe they're using a motor learning skill where they just learn right, left, right. And that's going to get me through to the end. Or maybe they're using a visual spatial skill. They're saying, okay, I'm in this lab, this weird thing. Oh, over by that door. I just need to work my way over toward that door over there, and that's how I will get through. Or maybe it's just simply using a search pattern. If I just simply keep walking, I'm eventually going to hit it. And I can become efficient. I can have a search strategy. I know if I always do this same thing over and over, I can get through it. The old, there's one of those Greek myths about that. If you walk through a maze with your right hand touching the wall the whole way, you'll eventually get through any maze. So, yeah. If you just touch one wall with your hand and walk through the maze, because basically the maze is inside of a circle. So if you just keep walking and you keep your hand touching, you'll eventually get through it. It might be the longest possible way to get through the maze, but you will eventually get through it. But the thing is that you can use multiple strategies. So say you were before you were using the left, right strategy to get through. And then I go and you have damage that inhibits your ability to form motor memories. That's okay. I'll just use something else. I'll compensate for that. I'll use the visual spatial strategy, and I'll just work my way toward the door instead. So you have to be very careful of whatever kind of testing that you do to try and assess for what the behavioral deficits are post-damage, that they are very specific to the brain region, and there aren't different ways that you can solve the task using different brain areas, because then you possibly won't see that. There's this study I read years ago, and I've had trouble finding it after I read this. But the theory that they were testing was that so in animal literature, older animals have poorer spatial memory abilities than younger animals. And so they were taking this to a real-world test, and they were going to measure how long it took people to find their cars in a parking lot. And so they were going to look at young people versus old people. And so according to that theory, older people should take longer to find their cars, remember where in the parking lot their car was located, than younger people. Now, when they did the test, they found that old people actually found their car faster than younger people. Okay, what's going on here? That's the opposite. Well, what they found out as they tried to figure out what was going on here, older individuals tended to use a strategy. They always parked in the same place when they went to the store or whatever. They always parked and so they didn't have to think about where their car was. They just went there. Whereas the younger person wouldn't bother with any kind of strategy and would walk out there and have no clue where their car was. So that was there. So basically, it was not a test of spatial memory at all. They thought that was a test of it, but the behavioral result they were seeing actually wasn't testing. It was testing whether or not you use a strategy when you park or not. So there are other ways. There are ways to compensate. There's ways people can do things, and that's fine. That's great. That gives us a lot of ability. So therefore, when we do suffer a brain injury for whatever reason, then having these abilities to use different ways to solve a task, ways to use different brain tissue to do old tasks, or to pick up novel pathways, or to strengthen other pathways. This is what allows us to still be able to function. So we might still have the same or at least a similar behavioral output, even though our brain is actually attacking the problem in a different way.